Okay, so last time we talked about electrical potentials, which are the resting, graded, and action potentials. And these are three kinds of electrical states that a nervous system cell can experience. And as I mentioned before, these are all states that basically just involve the cell itself. So it's kind of talking to itself, okay? Now we're going to talk about two ways that nervous system cells can talk to each other, or chemical signaling in the nervous system, okay? Chemical signals or signaling in the nervous system. Okay. The first one we've already talked about a bit. It's called the gap junction. Yes. Okay. Okay. The gap junction. So if you remember, back when we were talking about glial cells, and we were talking about satellite cells that cover ganglia, okay? So those satellite cells, their membranes, they just kind of overlap with each other. It's like they're holding hands. And now imagine, if by holding hands, pores appear between your skin and chemicals from your body pour into the other person's body through your hands, which have these pores. Okay, anyway, gap junction. That's what that is. And remember, it looked a bit like, so if we have two different membranes, so if I have my first lipid bilayer, oh sorry, I did this backwards. Okay, there's the first one, and here's the second one. Oh, it can be closer. Oops. Okay. All right. Two cells. And then we'll put the gap junction protein. So it's a structure of 4 proteins usually that are overlapping each other just enough to create a pore or a hole in between them where ions can pass from one cell cytoplasm. Okay, maybe you already know this one. Cytoplasm to the next cell's cytoplasm. Okay? You can continue drawing the membrane on the other side here. Okay. Just to show that it's just embedded into and connecting the two cytoplasms, okay? So that was a gap junction, and our main example of cells that use a gap junction was the satellite cells, the glia that surround the ganglia, mostly along our spine, okay? So then the next one is the synapse. Okay, so let me see. I'll just move over here because we're gonna have a lot to say about the synapse. Synapse. Okay. So a synapse is what forms when the cell membranes of two cells get very, very close to each other without touching, okay? So, forms when the cell membranes of two cells come very close to each other without touching. Okay? So unlike the gap junction where they're touching, okay? But here they do not touch, okay? And both graded and action potentials can trigger the release of chemicals in this synapse where they come really, really close together. Action potentials can trigger the release of chemicals in the synapse. Okay. So if we have our familiar multipolar neuron, it's just doing its thing. Okay. Whee! All right. And then we have our second multipolar neuron who happens to come very close without touching. Oh, I can't touch there. It's on a very, very tiny scale. That one goes off and away into the distance. Okay. And let's not forget, this is probably got some kind of myelin sheath on it. Okay, cool. So if we zoom in on one of these right here, okay, I'm going to blow this up. All right, nice big box to draw our synapse in. Okay, so let's see. We've got a membrane coming from the top. Okay, so it looks kind of probably like this. Okay, and we've got another membrane coming here. Okay. All right. So we have here a vesicle waiting. Remember what a vesicle is? A vesicle is a little bag full of chemicals that was generated by this cell all the way up here and then transported down to the axon terminal where it's now waiting. Okay, so we've got a little bubble full of chemicals. Okay. All right, so when an action or a graded potential fires enough to change the electrical potential down here at the axon terminal, we have what are called voltage-gated calcium channels. Okay, so let me draw this is a voltage gated Ca2 plus calcium channel. Okay? All right, so when a nerve impulse or a spike or an action potential, when that arrives here, what's going to happen is a bunch of calcium is going to now enter into this cell. So, what is it? I'll just do this. All right. This opens up, and now there's lots of calcium ion. Lots and lots of calcium ion inside of the cell membrane. Okay, so what does this do? This tells the vesicle, this tells the vesicle, oh, I need to go and release the chemicals into the synapse. Okay, so our vesicle is going to now move down here, and it can do one of two things, okay? So the first thing it can do is, let's say this is number one. The first thing it can do is it can basically, what is the term? Meld itself. It can attach itself to the cell membrane and just like open itself up and release the chemicals into the synapse all by themselves, okay? This one is called exocytosis because it was inside of a cell body, a little site. All of these, remember how there are a lot of cell names that are something site, astrocyte, oligodendrocyte. So the site is like, I'm a cell body, and exocytosis is to release something out of the body, okay? So this is, okay, I'll do it like this. Exocytosis. There we go. Okay, the next thing that can happen is the entire bubble, the next possibility, the entire bubble can decide to escape out of the cell, still holding on. Okay, and then it will, guess what? Merge with the other side. So this is now going to go all the way across. So let's give it a bit more scale. And then it releases it directly into the cell. So that is also has a name. Okay, so let's label a couple of things. I'm pretty sure this is called endocytosis, but I don't have it written down in my notes, so I'm not sure. You will have to fact check me on this. I'm pretty sure this is endocytosis because you're shoving it into the cell body as opposed to forcing it out of the cell body. So we'll go ahead and label this to endocytosis. And everybody is responsible for fact checking me on this, okay? Is this correct? If it's not, what does this word mean? Okay. All right. So let's label a couple more things. This here is called the synaptic cleft. Oh wait, I don't want to label it down there. I don't want to lose that space because we're not quite done talking about the full life cycle. Synaptic cleft. Okay, so these chemicals, they're going to interact with some of the membrane proteins on this cell. Okay, and now I'm going to label them. This is where it all started with the nerve impulse coming in and letting a bunch of calcium inside of the cell and activating a vesicle to go do something with the chemicals inside of it, okay? So this means this is the pre or before the synapse, presynaptic cell, okay? Which means this one is the postsynaptic cell, after the synapse. Postsynaptic cell. Okay. So, did I label everything? Yes. Okay, yes, now I get to go to my favorite part, which is obviously the glia that's involved here. So when this chemical is done interacting with the membrane proteins of the postsynaptic cell, what's going to happen to it? It doesn't just suddenly puff out of existence. So what happens is there are some glia who have probably sent their feet here. Okay. The nucleus, like an adorable little cyclops eye. This is not really to scale, but anyways, Here is our astrocyte. Astrocyte, buddy. Astrocyte. And what they're going to do is they're going to collect the chemicals. They're going to reprocess them, recycle them, and then feed it back to the neurons. We'll feed the nutrients back to the neurons so that they can make more of whatever chemicals that they want. Okay? So the astrocyte is the final step of this cycle. So electrical activity arrives to activate voltage-gated calcium channels, and that lets in a lot of calcium ions, so a lot of positive charge, which activates the vesicles, which are little bubbles, little sacs full of chemicals made by the neuron or the glia. And the vesicle is now going to travel towards the synaptic cleft. Okay, that's where the two membranes are the closest. And it's going to either merge with the cell that it's already a part of and release the chemicals into the synaptic cleft, or it's going to pop out, go all the way across the synaptic cleft with its chemicals, and then merge with the next cell, the postsynaptic cell, in order to release chemicals. Okay, and this release of chemicals can either create some other chemical reaction inside of the cell, maybe it changes the proteins that it's making, or it can also activate an electrical potential. Okay, it can by allowing for the influx of ions somewhere over here. Okay, so let's write that down. Color, what color? Let's use purple. Okay, so once the chemicals arrive, either way, whichever one it's doing, both of these situations, they're going to leads to either a chemical reaction or an influx of ions. Okay, that's usually how the signaling goes. Some sort of ions moving across the cell membrane. Okay, I think that's it for the synapse. And next, okay, we've been talking a lot about these chemicals. There are chemicals that the neurons create, that glia create. They package it up, they send it along, and then they pass it to each other. Well, what are these chemicals, and what do they usually say? So next we're going to talk about major neurotransmitters in all of the different areas of the nervous system.